Dr. Suthesh Sivapalaratnam with the ISTH meeting in London. He was a key member of the local organising committee. He has a big clinical practice in haemophilia, one of the largest in the world, and he looks in particular after the paediatric service, and he's genomic lead at Barts. He's also got a company. He's chief medical officer of a company called Blood Counts, and he's done a number of consortia. He's got a number of grants, and he's got a number of publications, but he is particularly well qualified to talk about thrombogenomics. So thank you very much for coming today.
Thank you, Beverly, for the very kind introduction. And it sounds all very impressive, but compared to you, that's obviously still nothing. Thank you, everyone, for listening to the talk. And here is a nice view of, if you look out of the day unit at the Royal London, where I'm based a lot, and you can see on one side the city coming close to us. But also in this picture, you see very closely our biomedical campus, and I'm very fortunate to be part of that development. So today, I'll be talking to you about thrombogenomics. So what are the developments in genomics and genetics in thrombosis? And there's quite a few excitements going on. I've been in the field of genomics for quite some time now. And it's really nice to see how the kind of academic and scientific part is coming close to clinical practice. I'm very fortunate to be one of the consultants at Barts, and I hold an affiliation at QMUL. And a lot of the science which I'm presenting here has been made possible by the NIHR. So what will I talk to you about? So I'll talk about a little bit of the background of thrombosis. This is things that you probably know, but just to land you softly. Then I'll go into genomics in thrombosis. So a lot of the things in thrombosis and the traditional risk factors were discovered through genomic research. I'll talk to you about the latest genome-wide association studies and the resulting polygenic risk scores. And I'll also talk to you about how this could implement our clinical practice and where it could be useful and where more work is needed. And finally, I'll show you some data on a really exciting recall study we're doing, and it's a bit of a plug for the study, hoping that you might be interested in contributing. So as you all know, there are numerous risk factors for thrombosis, and the standard risk factors are age, obesity, family history, obviously a previous DVT, recent surgery, hospitalization, and prolonged immobility. But now, recently, we also know that cancer can be a risk factor, chronic disease, and as recently shown, obviously, with the pandemic, viruses such as SARS-CoV-2 have been a major contribution in the risk for thrombosis. So these risks can also be quantified. So one of the major studies, which, as Beverly said, the UK has been on the forefront of, is the UK Biobank. So the UK Biobank is a large cohort of men and women between the age of 40 and 69 years who have volunteered to do this research. And this cohort, this prospective cohort, was initially recruited between 2006 and 2010, and it contains about 502,000 people. And if you look at this cohort, this is data published in Nature Scientific Reports, you can again see the different adjusted hazard ratios for venous thrombolism events. And some of the risk factors, like we discussed before, are here. So you can see that age is a risk factor, and with an increase in age, there's a higher risk of thrombosis. You can see the factors like BMI. So again, a high BMI of between 35 and 40 seems to be a clear risk factor for venous thrombotic events in this big cohort, and other things like smoking come quite clear. So here you can see the risk by hazard ratios up somewhere for the major factors between 2 and 4. As I said, family history is also a clear risk factor for thrombosis. And now, because I talk about genetics, it's very easy to just assume that that's all shared genetics. The estimates are that genetic risk factors contribute to 30 to 60%, but I'm very much aware that that will be shared environment and shared environmental factors. And some of the factors we discussed previously, like lifestyle, BMI, smoking, unfortunately also cluster in families, and part of that will be genetics. And if you want to pin that down with the number, papers published about a decade ago showed, and this is something we know, that once you have a sibling with a VTE, if there's one, your ratio compared to the baseline goes up by 2. If you had 2 affected siblings, your risk goes up by a 50-fold, especially if that's a young age. So as I said, that reflects the risks coming with families. So when we talk about genetics in general and genetics in thrombosis, how do we even study that? So the standard way of studying that has been family studies. And through family studies, we found in the past the quite severe disorders. Later on, about 5 to 10 years ago, we went on to candidate gene studies. And more recently, we've started doing genome-wide association studies. The technology for these studies has progressed massively, and met with progression of the technology, have we been able to type deeper and deeper. So when I started doing my PhD, about 15 years ago now, we were very interested if we could look at a few variants in candidate genes. Later, towards the end, the SNP arrays came, where we can type about 50,000 initially, but now about 800,000 genetic variants on one chip. That wasn't good enough for the genomicists, so we moved on to whole exome sequencing, where we can sequence the variants of the 22,000 coding genes, which also make proteins. And now we even do whole genome sequencing, and those can be used for association studies. But the big question remains, what is the usability of all of these technologies in medicine? And I think there is some usability. So I'll talk to you in more detail about polygenic risk scores, of which 2 studies have been instrumental, the UK Biobank and the Million Veteran Program in the US, and how we can use this knowledge in NGS diagnostic platforms. So again, when you look at this figure, so here on the x-axis, you see the frequency of an allele or a variant, and on the y-axis, you see the penetrance of the disease. So we started with the low-hanging fruits in genetics. So if you have a variant which has a high penetrance and is very rare, then that normally tends to run in families, and you see Mendelian disorders. So in the hemostasis perspective, for instance, Glanzmann's, Bernard-Triet's are like clear versions of high penetrance and rare variants. If you want to study common variants with a low penetrance, that's where the GWAS studies and the association studies were most useful. And now, as time progresses, we're going down and down the other bubbles. And it's also more clear that this landscape is not this straightforward. So the inherited thrombosis, we all know from a clinical practice, are protein C, protein S deficiency, antithrombin deficiency, factor V Leiden, heterozygosity or homozygosity, and the prothrombin variant. And a lot of these deficiencies and protein deficiencies were picked up by studying rare cases or rare families. We also know, if you look at this review from the New England Journal in 2001, that none of these variants are very black and white. Obviously, even in populations which are healthy, people will have variants in these genes, will have reduced levels, but as you go into patient cohort and enrich for thrombosis, the percentage and the frequency of variations in these genes and variants increases. So in a healthy cohort, as you can see, for instance, for a antithrombin deficiency is 0.02%. If you look in unselected patients with the thrombosis, it goes up to 2%. And if you then look in selected patients, and by selected, I mean young, relatively young thrombosis with the family history of VTE or recurrence, you can see that the frequency of variants and abnormalities in this protein increases. So that was how we looked at Mendelian disorders, but a lot in the past years have been around genome-wide association studies. So just to recap for those who might not know, genome-wide association studies are not complex. So on one side, you define a group of cases, and on the other side, you have a group of controls. And case control studies, we've done those forever. But what we do in the genome-wide association studies is that we type the cases and the controls. And by typing, I mean, we look at their genetic variants. And as I said, initially, we could look at maybe 50,000 genetic variants. But now we can type at a higher resolution. So we can look at about 800,000 genetic variants in cases and controls. And then the statistics are relatively straightforward. It's a chi-square test to look, is a specific variant associated more with disease or health? And that was the basis of genome-wide association studies and still is. But what has changed is that with more and more of us organizing ourselves well, we've had bigger cohorts, which are better phenotyped clinically. Because of that, we can detect smaller changes. And also the technology of the platforms has improved. So where we had SNP typing, now we have whole genome sequencing, but the mechanism hasn't changed. So the really interesting thrombosis GWAS, the most recent two are one by the UK Biobank. And as a reminder, the UK Biobank had about 500,000 healthy volunteers there. And a part of these 500,000 would have gone on to develop a thrombosis. So in the UK Biobank, they tried to make a clinical risk score for the development of thrombosis. The biobank was also fully typed on an FE800K chip, but here we just look at the clinical risk to develop thrombosis, and they could make three categories: low, intermediate, high. And the difference between those categories was a fourfold if you just look at the risk of developing thrombosis based on the risk factors as age, sex, BMI, previous cancer, smoking, and fracture risk. And these risk factors were picked because those were the most prominent in the analysis. If we do something similar for this, the variants, so out of the top GWAS variants of the association study, out of the 102 top variants, we picked the 36 variants which were most indicative. And again, we made a stratification of low, intermediate, and high. And there you see the difference between the top and the bottom risk categories is a threefold. But the power is in combining it. So when you combine the clinical scoring, and why wouldn't you combine it, and the genetic scores, you can see that you can better stratify people who will develop VTE, and the difference between the highest and the lowest group is 7.5. So again, the combination of genetics and clinical data can help risk stratify, and I think that will clinically be very useful to further prevent thrombosis by picking those who would benefit from thromboprophylaxis. Genomics is like expanding. So after having done the UK Biobank GWAS, the Americans combined with the UK Biobank with their Million Veterans project. And by doing this combination, we suddenly, for the association study, had 26,000 cases and 624,000 controls. And the findings of these genome association studies were replicated in an astonishing 17,000 cases and 176,000 controls. Discovery and replication are standard in these studies. And the latest, the big paper in Nature Genetics, identified 33 loci, so 33 regions of the genome which are associated with thrombosis, of which 22 were new. Now, these loci each warrant further investigation, and that can take up to decades. However, where I find it clinically interesting is that you can do the same game. You can make another polygenic risk score. So this polygenic risk score is based on the Multi-Million Veterans Project and the UK Biobank. And let me just talk you through this, because people get scared, but actually it's not scary at all. So you see a Gaussian curve. You could have drawn any Gaussian curve for any trait, for height or possibly, but this Gaussian curve represents the polygenic risk score for VTE, to develop VTE. And as you can imagine, there will be extremes. Now, if you look at the highest group, the highest 5% of developing VTE based on the polygenic risk score, we can look in that section into more detail. So this highest group are those who either have a factor V Leiden variant, which we knew that's something we accept, a prothrombin variant, and you can see the odds ratios in the bottom here. But also, what is quite interesting is if you make a combined stratification of the top scoring SNPs or variants, you get an odds ratio which is very comparable to those two. You can strengthen this risk score by making all kinds of combinations. So combinations of prothrombin variants with these SNPs, or the factor V Leiden with these SNPs. So why am I showing you this? I'm not showing you this because I think that's massive odds, but I'm just showing you that we can incorporate some of the state-of-the-art data from the GWAS studies to better predict risk. So that brings me to the current state of play in thrombosis and the genomics. So in red, you can see the kind of traditional genetic risk factors, which were all protein-based and coagulation cascade-based. But since then, I hope I've explained to you that we generated another 33 loci, which definitely need further study. Now, you can ask, where does this bring me clinically? And for that, I just want to take you back into one of the big genomic projects which have been led from the UK, and that was the rare disease pilot, and that was the start of the pilot for the 100,000 genomes. And in that pilot, over 13,000 participants partook from all over the world, and they all had whole genome sequencing. And the goal of this was to understand and find those rare disorders, but also to understand and learn how we would report variants in a clinical suitable way. And that arm was done by Thrombogenomics. So Thrombogenomics was again an international collaboration led very much by the UK, and people like Andrew Mumford, Professor Mumford, Professor Chowdhury, Dr. Gomez have been instrumental in this. So in Thrombogenomics, what was new is we tried to do a panel test versus testing a single gene for disease. And why would we do that? The benefit of doing a panel is that you can test multiple genes in one go rather than sequentially doing one gene. It becomes cheaper because if you have to do a gene every time, that's just quite time consuming, and it is more affordable. And through work done through that pilot, we now have NGS testing in clinical practice for hemostasis and thrombosis. The base of this Thrombogenomics platform, the capture, was that it would contain over 100 genes. It would contain all the protein coding regions, but also the beginning and the end bit of the genes, because those have regulatory elements, and all those areas which we thought would be implicated in disease. And this platform, it's kind of a virtual thing that changed over time. As I said, it's not only for clotting, so it has platelet genes, which are obviously involved in bleeding disorders. but it also has a list of pro-thrombotic genes. And this list is about 17. And these are the lists which were curated by also the ISTH subcommittees as most important in clinical practice. So this is a beautiful tool from research to clinical practice, and we all have access to this panel. We validated the panel, so we looked at 2,396 patients with a bleeding or platelet disorder, and we looked at how the panel performed against their normal standard test. And if we focus on the thrombotic disorder, which was about 10% of this cohort, in about up to half, we were able to identify a variant. Some were pathogenic, so we know that they were causal. Some were likely pathogenic. They might be causal, but it needed more information. And there were also variants of unknown significance. But in a good half, we never found a variant. So this could be because it wasn't genetic or we weren't there yet. And I think that these panels should be incorporated in diagnostic pathways. And we do that on the other side, so on the bleeding and platelet disorders. And why do we do that? Because actually, it's a slightly simpler disorder. So some people bleed, some people have low platelet counts, and it can be underlying to a lot of different genetic variants. And you can see here that platelet disorders, sorry for the sidestep, is a multisystemic disorder. And we better know that certain platelet disorders, for instance, are result in renal problems or in cancers. And that's why we want to know what the underlying molecular basis is, because it impacts their treatment and management. So coming back to what do we now do with this R97 panel? So this is the NGS panel, which is available. Now, the latest BSH guidelines written in 2022 do mention this panel, and they say, well, you can use it for thrombophilia with a likely monogenic cause. So it has to have clinical features. And as I spoke to you through the talk, you can see young age, spontaneous, very weak environmental risk factors. You could do this test, but it says very clearly, testing should only be used where it will impact clinical management. And that's the challenge if you follow the guideline. Because what do they say? What does the BSH guideline say on thrombophilia testing? Well, my summary is actually do not test. And I understand that with the clinical hat, but it's quite difficult with the scientific hat. But what they say is if you do a test, that should result in an effect for your patient. And just testing for the sake of it will not help anyone except for the scientific community. There are a few exemptions where they say asymptomatic first-degree relatives of probands with protein C, S, and antithrombin deficiency, where this might influence the management and life choices, can be tested. So in an ideal world, we would have this beautiful loop where we do clinical data, genomic data, and if we know what the variant causes, by an MDT, we report back to the patient. And if we don't, we do genetic analysis. Unfortunately, in thrombosis research, we are not there yet, but I think we can be and we should be, because where do I think that genomic fits into the picture? So I think it would be to discuss who to give thromboprophylaxis. As I showed you, we have much more evidence about risk by polygenic risk scores. We should think about which women in pregnancy and postnatal periods should have thromboprophylaxis. We have beautiful guidelines, but maybe we can expand on that. We can talk about tailored assessment of duration of treatment. How do we counsel young patients who've had an event? What is their likelihood of a re-event? And could that be based on a genetic cause? Should we talk about prevention in high-risk families? There's not much data yet, but we can gather that as a community. And what about additional phenotypes? I mean, why would platelet disorders be related to other phenotypes, but inherited thrombotic variants not? So, having said that, I just wanted to let you know that I hope I've convinced you about the need for further research, and we have ways of doing that. So the NIHR recruitment is open for young thrombosis. So if any of you have patients, and this is for scientific purposes, who are young, under the age of 30, with a family history of thrombosis, please do send them to me, because we can look in more detail. And what we do there is we look at all the clinical data, we do whole genome sequencing RNA sequencing, proteomics, to align those things, because we believe that in a subset of the individuals where we can't find a monogenic cause with the standard panels, it was like 50% in the R97, it could be due to variations which cause changes in expression. So coming to my end point, I think genomics research in thrombosis has recently resulted in useful things, the NGS panel, GWAS loci, which we can use for polygenic risk scores. There is a role for genomics in diagnostics of thrombosis. The R97 panel is accessible, and I think we should start adding some of these polygenic risk scores to those panels. And the recall study is open for recruitment. So do please send interesting cases to me and I can talk to you about that. So without further ado, this work is obviously not just done by me, but a big panel. And I just specifically wanted to thank Keith Gomez for all the work he's done in thrombogenomics, Willem Ouwehand, who's been a lifelong mentor, but also new blood such as Amar, who's in the left top corner, who's been really grateful in gathering these families with young thrombosis. Thank you very much.
Terrific, Suthesh, and you've managed to finish just about at the right time, which is brilliant. It's just starting, isn't it? The whole of thrombogenomics is opening up. Can we just be clear? You want patients or you want their blood samples if they fit your criteria that you've talked about?
Yeah, so thank you, Beverly. So there are numerous NIHR sites, so contact me because it might well be possible that there's a closer site than myself at the Royal London, and we can even organize home visits for those patients. So yeah, we can definitely talk about that. But
you've got funding, have you? This is a funded research project. Yeah. We're talking millions here, are we?
Yes, there is funding for patients to be even visited at home to take their blood, to get the genome sequencing done, proteomics done, RNA sequencing done. There's a whole team ready to go, and we've recruited our first patient. So funding, should it be a problem, I can sort all the logistical headaches with my team. But just if you have an interesting case, just drop me an email and I can guide you through that. And there is a lot of scope, Beverly, for research. So we gather some more data than we can actually do proper research on, as you can imagine. So if people are interested in doing research within the community, that's also absolutely fine. I'm not planning on sitting on the data myself. It's too much to handle. So anyone is welcome.
And is there a website we can look at?
Yes, so if you go to the NIHR recall study, but I can put that in my slides, which I will share with the people who were here.
Okay, well, I have to wrap up now because we've got another session starting in 5 to 10 minutes. I want to say to the speakers, wow, thank you so much. I've learned such a lot from all three of you. And to tell everyone, if you came late or you had to leave early, this is going to be up on the Thrombosis UK website for you to see, perhaps for the first time, or just to revisit for the second time to catch up on all this amazing new information. So thank you all for fabulous talks, and let's hope we can do this again next year and have an update. Bye for now. Thank you.